All right. So we're going to continue on with the pressure flow today. I will discuss the exam once I've graded the essay portion, and then we can go over aspects of it, but it's going to be fairly brief in lecture. If you want to go over it in more detail, please set up an appointment with me, and I'd be happy to do that and come up with some ideas for perhaps some better study strategies if you were not happy with your score. Keep in mind that there are a lot of points yet to come in this class. So if you stumble on the first exam, it's not the end of the world. And that's why I don't make it a 100-point exam or above 100 points at the start, okay? So please, we can set up an appointment to go through it in detail. And I have plenty of ideas on how to study if you want to do a more effective study strategy. So please make use of that. I want you to actually learn this stuff. It's cool stuff. I want you to learn it. So, and it'd be nice to learn it beyond just, study, take the exam, dump it out of your brain. If you can actually retain it and use it, because I'm hoping at the very least you'll have plants in your life, in your house, in your business, in your apartment, wherever, and you will know how to deal with them so much better with these principles. All right, so we're shifting into pressure flow. So we're in the phloem instead of the xylem, but right next door, essentially, to the xylem is the phloem. So we've been working through the ideas of how they were trying to figure out the mechanism of phloem translocation, let alone what's in it. They got a pretty good handle on what gets translocated through the phloem, but then the mechanism took some doing. So before the exam on Wednesday, we did talk about this idea of they were starting to lay the groundwork where the data was coming in that it's a bulk flow process where there is some kind of pressure gradient that is the force needed in order to get the phloem translocation to occur. And so we're talking about pressure due to buildup of water to allow for a pressure gradient to develop. And that we see a pressure gradient from the source where there's higher pressure at the source and lower pressure at the sink due to how this process works. And then it is linked to what's happening in the xylem, because much of the water that's needed to build up that pressure is actually coming from the water in the xylem right next door, okay? So before the exam on Wednesday, we did set up the scenario of the process. Well, let me just recap that. And so here you can see the source. So a mature leaf, let's say, of a beautiful, maybe it's a maple tree. And you have the source cell that they're referring to here is just maybe it's a spongy mesophyll cell in the leaf, okay? And they're doing photosynthesis, so they're making all the sugars, which are represented by the red balls here. And then you can see the idea of phloem loading occurring in the leaf, where the sugar is getting loaded into the companion cell, which is the blue cell here. And then that can quickly get transported to the sieve tube element. Okay, so you get this buildup of sugars. So thinking about water potential, that means you're messing with the psi sub s portion of the equation. The minute you start putting more solutes into that system right there, that's going to drive your psi sub s more negative. More solutes, more negative. More solutes, more negative. Okay, so as soon as that starts to happen, that's going to shift your overall water potential to become more negative compared to surrounding cells. And so that will allow for the inrush of water flowing across the cell membrane from less negative to more negative. All right, so the water potential in the source is more negative than surrounding. And so you will get that water coming from the xylem coming into the sieve tube element. That's going to make your psi sub p go up, up, up. You're going to develop a positive pressure in the source. Okay, so that's one half of the story. That's just what's happening in the source. It really doesn't make sense until you tell the whole story and consider what's happening in the sink tissue. So let's do that next. So here at the sink, let's go back to the potato plant, because I like potato plants. let's say it's a developing potato tuber in the soil. So now if that source cell is the tuber, the flesh of the tuber that you will eat if you have French fries or baked potato. All right, so we have to get sugar into that. So now at the sink portion of the plant, we call it unloading, phloem unloading. All right, so we have to unload the sugars into the potato tuber tissue. So that's what you see here, where it came to the potato tissue area. The phloem is, the sucrose is leaving the phloem and going into the target tissue. So if you have sucrose leaving the sieve tube element, exiting that will mess with your psi sub S again. And that will make your psi sub S less negative because you're losing solutes now to that beautiful potato. So as the potato is saying, yep, I want that sucrose, I want that sucrose, you're putting the psi sub S in the sieve tube element closer to 0. It's becoming less negative. So that means your overall water potential will become closer to 0. It'll become less negative. So that means water will probably go out of that sieve tube element to a more negative water potential, which is typically the xylem right next door. So here you can see the overall water potential is negative 0.6 in the xylem. Over in the sieve tube element right next door, it's negative 0.4. That's less negative to more negative. 0.4 to 0.6, less negative to more negative. So that creates a lower pressure in the sieve tube element compared to the pressure buildup in the source, in that same, sieve tube element highway system. So you have high pressure at the source, lower pressure at the sink within that same sieve tube element route. Making sense? Kind of? So this is the entire picture put together from start to finish. This is exactly what we just looked at. I've just uncovered all the parts so it's all intact. So you have the buildup of pressure in the source, leaf tissue typically, higher pressure. So psi sub P here is 0.6. Psi sub P down in the sink by the developing tuber is only 0.3. So that's a pressure gradient. So you'll have bulk flow of the sugar that's dissolved in the water through the phloem from source to sink. Okay, there's something else on here that's a little odd. I'm going to come back to. All right, I'm going to come back to it. But let's continue on a little bit. So in words, this is what's going on. So I've been showing you a visual. Let me put it into words if you're more of a read-write person. This may help. So the spongy parenchyma cells, the palisade parenchyma cells in the leaves, they're busy doing photosynthesis, making that sugar, and they got to send the sugar to the nearest vein, to the sieve tube element. So CCSTE, that stands for companion cell, and sieve tube element. And we often think of it as a unit or a complex. So you dump that sugar into that vein, into the sieve tube element. And again, that's increasing your solute concentration. So that's messing with your psi sub S. So your psi sub S becomes more negative, which drives your overall water potential more negative, which will then cause water uptake, typically from the xylem right next door, because that will be a less negative water potential over there to a more negative water potential in the sieve tube element. And that builds up your psi sub P. You get a nice hydrostatic positive pressure at the source now with that inrush of water. So that's all happening in the source area, but you have to complete the thought. Otherwise, this doesn't make sense. So you have to tell the story how there's a gradient, not just, oh, there's high pressure in the leaf. That's not enough. You have to convince that there's a gradient happening to allow for that movement. So at the same time, in that same sieve tube element, highway, you will have sugar being unloaded into the source tissue. perhaps a potato tuber. So now the sucrose is leaving, it's exiting out of the sieve tube element down at the sink. So that's going to drive your psi sub S to be less negative. Your overall water potential starts to go less negative. And somewhere nearby, there's going to be more negative water potential, and it's usually the xylem. So then water will leave the sieve tube element, go from less negative water potential to more negative water potential into the xylem. And of course, if you lose water, you're going to lower your hydrostatic pressure at the sink. So your psi sub P starts to come down. Now you've completed the story where you can establish that you created a gradient, a hydrostatic pressure gradient from source to sink. So as long as you have that pressure gradient being maintained, that means you have to have continual loading at the source, continual unloading at the sink. As long as you have that going on, you'll maintain that pressure gradient. Questions on this idea? Okay, so we're just playing with the water potential concepts.
The companion cell, it has like a sucrose gate or something. How does it react to those?
And how does the sucrose get from the companion cell to the sieve tube element, you mean? It's usually through plasmodesmata, but not always. We're going to talk about that next, actually. Like, how does that actually happen? And there's some weird stuff going on with that, too. So, okay. So pressure gradient. So when we talk about water potential and movement of water, we're thinking about osmosis, right? And osmosis has a two-part definition. So it's the movement of water. What's the second part of that definition? Exactly, very good. Through a selectively permeable membrane. If you don't have both of those met, it's not osmosis. Keep that in mind now. Okay, so let's go here. And you've seen this. So, okay, there's a weird thing going on here. So I just told you that sucrose dissolved in the water is going to move from source to sink based on a pressure gradient. So we're talking about water movement. Now let's look at the overall water potential up high in the source. It says that it's negative 1.1 megapascals. Now let's go down to the sink and the overall water potential, eek, it's negative 0.4. So which of those two values is less negative? Okay. So I've just been saying to you all these, 3 weeks, water moves from less negative to more negative water potential. That's the law. And here it looks like it's not behaving according to the law because water is moving from the source to the sink, but the sieve tube element down there, it's less negative. And I've been telling you all this time, no, it's less negative to more negative. Here we're going more negative to less negative. Okay. You probably think I'm losing my mind. I wouldn't blame you. I feel like I'm losing my mind all the time. But osmosis. We say water potential laws come into effect during osmosis, which is examining water movement across a selectively permeable membrane. Once you consider the sieve tube element highway system, is osmosis being met, the definition of osmosis? When you consider the sieve tube element highway system, is there a membrane being crossed from sieve tube element to sieve tube element to sieve tube element? Remember, they stack on each other when you have these sieve plates.
These plates are orange membranes.
Exactly. I tried to say that way back during the anatomy lesson. No membranes here. It's all continuous, an open tunnel system through these sieve plates. So we do have movement of water and the dissolved sucrose, of course, but there's no selectively permeable membrane in the sieve tube element highway system. Well, there'll be gravity impacts a little bit, yeah. Yeah, there are gravity impacts that will affect this for sure.
So since there's an osmosis protocol, that the hydrostatic pressure gradient precedent?
Yeah. So really focus on the pressure gradient idea as you're thinking about movement. I mean, you still have to consider water potential ideas for the loading and the unloading. You have to think about that. But once you I tend to think of it as an internal highway system. Once you're in the sieve tube element, there's no membranes here in the sieve plate area. So from sieve tube element, sieve tube element, sieve tube element, no membranes. So therefore, it's not really osmosis in that part of the plant. Okay, so it's a little weird. I'm not going crazy as far as you know. So water moves by bulk flow. We're talking about bulk flow. No membranes are being crossed from sieve tube element to sieve tube element to sieve tube element. So it's a pressure gradient that you need to consider. That's what you need to zero in on, the pressure gradient. Is there a pressure gradient from source to sink somewhere in that plant body in that particular sieve tube element highway? So we haven't violated any standard laws of osmosis. So this took a long time for the plant physiologists to figure this out. This was no easy feat, especially with the limited technology, but it's still the most widely accepted theory. They actually call it a theory, not a hypothesis. And Dr. Bunch gets most of the credit for it because he's the one who kind of firmed up the concepts and did a lot of heavy studying on this. So they've looked at it. They've used confocal microscopes where they watch, because you can do live tissue samples with that type of microscope. We have one in the biology department. And so you can actually have living plants and watch the flow of phloem using confocal. And so they've done these types of work to actually get the experimental data to, come up with their idea, their hypothesis. And so they have real data. This is just sieve tube element, SE. So they're showing two tube elements here, and then there's different things that are in there. They're living cells, so there's different things that are showing up in the notes section. It explains what all the arrows are pointing to. But the big picture idea of this, believe it or not, this staining technique did demonstrate there's no membranes. I know it's hard to see with an untrained eye, but the sieve plates were clear. There was you could see the flow going without any membrane. This is a little bit better image. Again, just trying to collect the data to demonstrate there are no membranes from sieve tube element to sieve tube element near the sieve plates. There's nothing going on there. That's an open system from one cell to the next. They also had to rule out that this was something that needed ATP. They needed to think about that because that certainly would impact the physiology of the plant. If the plant had to invest a ton of ATP to get this to happen, they'd be exhausted. Thank goodness it turned out not to be the case. So this data is showing that ATP was not needed. So what they did here is they took a plant sitting at slightly elevated room temp, and then they were measuring the Phloem translocation. And then what they did was they cooled down the plant quite dramatically. And when you do that, ATP production gets way slowed down. And you can see there's a slight bump or decrease. It took, basically this is the plant going, yeah, it's cold. And it just took a little while to bounce back from that, but not very long at all. And it did bounce back 1 degree. It's like, oh, I can handle this. And it acclimated and then kept going with Phloem translocation. No ATP was needed. And so again, this was just data trying to demonstrate that you didn't have to have ATP for Phloem translocation to occur. So lots and lots of experimentation went into this to solve this mystery. And then, okay, what we've just been talking about is we focused on the sieve tube element. But of course, they're very curious to see, well, okay, so you have the palisade parenchyma cells, you have the spongy parenchyma cells, they're all doing photosynthesis, and somehow that sugar has to get to the veins. And how does that happen? So there was a lot of thought into that. And when they look at the fine anatomy of leaf tissue, leaves are just amazing, because when you look at the venation pattern, it is, on average, you can say that there's just a maximum of 3 to 4 cells before you hit a vein. And so what that's basically saying is leaves are very well, was it innervated? I can't remember the exact spelling of that word, but it just means that it's just really good venation pattern, very good micro veins, so that you don't have a ton of cells in the way before that sugar can end up into a vein. So of course they were looking at, okay, so we know that sugar production is, well, photosynthesis occurs in the chloroplast of each cell, but then you have to, and you don't know this yet because we haven't gotten into the details, but after they make some primary sugar products, that gets put out into the cytoplasm. And then that's where the sucrose gets made. So then the sucrose is in the cytoplasm, and now it's got to get from, like, let's say it's a spongy mesophyll cell or parenchyma cell, it's got to get from that spongy cytoplasm into the vein nearby, 3 to 4 cell distance maximum. So they thought, well, it kind of makes sense that it would go via the symplast. That seems, if it's being made in the cytoplasm, and we know that, so symplast movement of water or sugars, which movement, how is that happening? The plasmodesmata. So those are the internal tunnels from cell to neighboring cell. So if sucrose is getting made in the cytoplasm of one cell, it makes sense that it would just move through the plasmodesmata into the next cell, and hopefully it'll get, quickly to the vein. So that was a top pick. They're like, yeah, that's probably what's going on. But then they also had to consider other movements. And they said, well, could it be that somehow the sucrose is getting into the cell wall space and somehow getting translocated and dumped into the vein that way? So they had to look at that. If that happens, and they do have experimental evidence, there comes a point where it has to get actively loaded into the sieve tube element, companion cell complex. Anytime you say actively something, in this case, actively loaded, they're trying to get you to think about energy. Energy needs to be expended. And so they're saying, in this case, ATP is needed to take the sugar from the apoplast and get it loaded into the sieve tube element. I'll show you some visuals of this too to help. So that brings in the concept of PACT and SAT, which is primary active transport and secondary active transport. And I'll get into that as well. So the fancy word for that we're going to see, the details, is called symport. It's a symport process. You'll get sucrose and a proton moving together. So it's a symport method of sucrose loading.
Is this how sugar is get past these sucrose cells? Or I know when you're talking about water when they do the apoplast.
Yeah, exactly. Yep. We're forcing it. This is forcing it to go across the membrane right here. Yep, you got it. So here's a mock-up of a leaf in cross-section where you have the xylem and the bigger pebble cells here. Here's phloem in the companion cell, and they're just color coding it because they're going to blow this section up. So here's the spongy mesophyll here, palisades up top. So you have to imagine all these cells are doing photosynthesis, and then they got to get the sugar to the sieve tube element, to the vein. All right, so here, these are just some of the options that they imagined happening. And then of course they had to go dig for it experimentally. So here you have the, let's say it's a spongy parenchyma cell. It's taking in CO2 and through the process of photosynthesis, making sugar. And then it's using the the plasmodesmata, the symplast method, okay? Internal tunnels between neighbors. And that seems logical. It's like, that's easy. It can transfer from cell to cell quite easily all the way to the companion cell, which then can load it into the sieve tube element. So that's pretty straightforward. And that was, like I said, that was like their top pick. But then they said, well, they could in theory, see how it might happen at some point in the process. It might go apoplastic, into the cell wall. So here they're showing the same scenario. Initially, it's symplast, 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 and then here they're showing it crossing the cell wall at the phloem parenchyma cell. So right in the tail end there of the transport. And it's right there now as you get the sugar going apoplastic. Well, now it's got to cross the cell membrane in order to get into the companion cell. And that creates an issue. So in order to do that, they have to go through primary and secondary active transport. So that's kind of a pain. So that's what this is. So here they're showing if you have an apoplastic movement of sugar, you have to get this scenario set up where active transport means you're expending ATP. So primary active transport is being shown here by the expenditure of ATP. So the top half of that image where they show ATP is being used to push protons across the membrane, that is the definition of primary active transport. I'm hoping I'm jogging your memory and you've had this in some other class. This is basic chemistry. So primary active transport, I like to think of it as setting the stage. You're setting the stage for something good to happen. So what is the setting of the stage? You're creating a proton gradient. So here you see ATP is being used. You need that energy to push protons across the membrane. And so if that keeps happening over and over and over, you get more protons building up in the cell wall space there compared to what you have in the cytoplasm. So then you get a proton gradient. So that's awesome. If you can create a situation where you get a proton gradient, that's potential energy that you can use to do something with. In this case, they're going to do secondary active transport. So they're going to use these protons. Now, protons are going to want to diffuse across the membrane by the laws of diffusion. So what happens is it provides the necessary oomph to get sucrose dragged along with it. And so here you see the proton and the sucrose coming back into the cell on the same transport protein, coming through that same protein. So it's like the protons helping the sucrose get across. Like, here, come with me, I'll escort you across. All right, and so it's through the same protein. So that's the symport process that was being referred to in the previous slide. So all that has to happen if sucrose ends up in the apoplast. You got some work to do to get it into the companion cell and sieve tube element. So extra effort, but they can demonstrate, at least when they go experimentally and they look for it. This table, in the big picture idea, you don't have to like memorize any of this. What this is just trying to show you is that the transport proteins necessary for all of the primary and secondary active transport should be present in the pathway to get into the sieve tube element. We should be able to find them, otherwise it's not just going to happen by magic. You got to find evidence. So they were looking for, especially the symport proteins. They were looking for those translocator symport proteins, and they found them. So this table is just saying we found them. All right, so they have experimental evidence that they found the genes that code for those proteins. These are the actual transport proteins that were made. So enough different plants were sampled, and they feel confident that this is a potential method. So apoplast can happen, but it's, an extra investment of energy. The symplast method seems a little bit more straightforward, because since sucrose is being made in the cytoplasm, just keep it in the cytoplasm. Just keep going through the plasmodesmata, like this top picture here. So there's evidence. They can show that symplast movement does happen. So does apoplast. And here's a good picture of what they did here is radio labeled the carbon dioxide, fed it to the leaves, and then the leaves do the photosynthesis, and all the black is the sugar resulting from that. So that's radio labeled sugar. And what this is just trying to demonstrate is that all the spongy mesophyll and the parenchyma or palisade parenchyma cells, all of those cells in the leaves, they're all doing photosynthesis, all making sugar, and all of that sugar has to get loaded into the closest vein. So that's a lot of sugar getting dumped into the vein, and that's why all of the vein tissue has popped black. And she's saying there's a lot of sugar. So what that's implying is that there is a lot of sugar in the vein because so many nearby cells are all dumping their own share of sugar into the vein. So you get high levels of sugar in the vein, relatively lower levels of sugar in the palisade and spongy cells. So that made them go, well, wait a minute. If there's so much sugar in the vein, let me blow this up. Can I do that? Are you gonna let me? Oh, it's still in drawing mode. Sorry. Erase. Oh, erase. Come on, eraser. My computer has to think for many seconds before it will switch and do what I tell it to do. Where am I going? Oh, I wanted it off. So let me blow it. Come on. No, now I'm in erase mode. All right. So imagine all the spongy parenchyma, palisade parenchyma, they're all doing this and all loading sugar into the companion cell into them. And it's probably favoring the plasmablaton syntax movement. So you can imagine where lots of sugar. These are open tunnels. So if you have lots of sugar, you can imagine where there could be many times during the day where there's really high sugar in the companion cell. Why doesn't it just diffuse down into concentration gradient back up, right? I mean, tons of sugar all coming from nearby cells. You get really high, relatively high concentrations of sugar. So delta C over delta X by Fick's first law of diffusion, why don't you just have sucrose flowing out of the companion cell back into the spongy or the palisade? Well, I mean, this was really confusing to the plant physiologists. Like by law, it should backflow. Certain points of the day, you're going to get probably a lot of points during the day. You're going to get backflow, but they couldn't experimentally show that. They went looking for it, and they're like, not happening. Do plants know how to break the law of diffusion? Are they getting around it somehow? So I always imagine at this point in their experimental process, they went to the nearest pub and drank a lot of beer because they came up with a theory that sounds really outlandish at first. But when you start thinking about it, it's like, oh, yeah, that could work. So I always imagine they did this over some beers and a bunch of BS. But this is what they came up with. The polymer trapping model is what they call it. So over some beer and maybe some fish and chips, they said, okay, what happens if we get sucrose loading into the companion cell tube element complex and it doesn't stay as sucrose? It gets transformed through a chemical reaction into something else. So that's what's being shown here. Sucrose is getting a galactose molecule added to it. Well, that makes it bigger. Hey, what's sucrose again? It's a disaccharide. What are the two sugars? Glucose and fructose. Okay, so now they're saying, let's take that glucose and fructose, let's add galactose to it. That's going to make raffinose. Let's add another galactose to it. That will make stachyose. We had a slide that was going on and on about that earlier. Okay. And then they said, so if you keep adding galactose units to sucrose in the companion cell or tube element area, it would make it too big, so it physically couldn't fit back out of the plasmodesmata tunnels. It's just too big. Simple, right? But kind of weird. I remember the first time I learned this, I'm like, that's wacky. That's why I always pictured people drinking heavily through this part. Like, you gotta be kind of, think outside and be really creative. And so they said, if you keep, adding some galactose units, you would make that sugar molecule too big. And then it couldn't fit, even if it really wanted to diffuse back into the spongy mesophyll. Physically, it just couldn't do it because the plasmodesmata diameter would be too narrow. There, we're done. So they drank their beer, went back to the lab, and hopefully made it through the experiment without any major accidents. And they said, let's see if we can find the experimental data to support our wacky idea. So they worked on that for a while, for probably several weeks and months. And so they had to look for presence of these other sugars. They had to look for they had to measure diameter of the plasmodesmata. Both the plasmodesmata connecting like the spongy mesophils, and then once it gets to the companion cell, let's measure those. And so we're going back to this slide where we have sucrose, which is the glucose and fructose, but you keep adding the galactose units and it physically gets bigger. And so it should physically be impossible to diffuse back if the diameter measurements work out. And so here you see bundle sheath cell, which is just the ring of cells around the vascular tissue. So you have to imagine there's spongy mesophyll off to the left here, let's say. So there's spongy parenchyma cells, and they're making sugars and then getting them in here. Well, then here you can see the artist drew the plasmodesmata from the bundle sheath to the companion cell. The intermediary cell, that's a companion cell. It's just a special type. So this is the blue one in the middle is a companion cell. So you look at the width of the diameter here of the plasmodesmata and then compare it to the diameter loading into the sieve tube element. And you can see how the artist drew it. There's a difference in diameter. So they had to experimentally look for that and do some really careful measurements. The other thing they were looking for is, are there the proper enzymes and sugar molecules to allow for these reactions to take place once sucrose gets into the companion cell sieve tube element complex? Are the enzymes there to do all the building of the galactose reactions? So they looked for both of that and they found it. So their wacky idea still holds today. The polymer trapping model. You've trapped the polymers, the sugar polymer. It's no longer sucrose, it's a polymer now. And it's trapped in the companion cell. It's not physically diffused back, even though there's a concentration gradient many times through the day where it wants to diffuse back based on the laws of Fick's first law of diffusion. Have I convinced you? I just think that's funny. So here, this slide is just put in for study purposes with all the extra words. So as you're studying through it, hopefully that will convince you. So they found the evidence for their polymer trapping model. So good on them that they came up with that idea and it worked. It's always nice when one of your first outlandish ideas actually hold up. This again is just data to convince you there is data to support this idea. It's not just some people saying that. And so again, you don't need to memorize any of this. It's just to prove to you that they did go and take the experimental measurements and collect the data, and they could find the necessary enzymes and proteins, and they measured the plasmodesmata width of the diameter. They did all that. Okay. All right. So we solved that riddle where you can still get movement of sucrose all the way, continually going in kind of one direction, loading it into the sieve tube element, even though all those cells might be loading sucrose into the same vein. You can still get total movement into the sieve tube element, no diffusion out of it. Now we have to think about the unloading process. And let's use the potato tuber tissue as an example. Now this one I'm not going to go into as much detail because there's several pathways that they have discovered and they have the data to support it. So I'm going to give you the big picture idea. And basically it comes down to what sink tissue are you thinking about? That makes a big difference. And then based on that, they've discovered many different unloading methods. So you have to take the sucrose in the sieve tube element at the sink and unload it out of the sieve tube element, out of the companion cell, and get it into potato tissues, just a bunch of parenchyma cells that are really good at storing up starches. So you have to get it into the parenchyma cells of the tuber. So they know experimentally they can demonstrate that it's symplastic, and they've done a lot of work with this with root tips, how sugar gets to the growing root tip. Of course, root growth and development is hugely important, as we'll learn, because the next topic will be all about roots and how they have to absorb water and minerals from the soil. So you need a good root biomass. So Symplast certainly works in unloading, but they've also discovered apoplast, particularly with seeds that have those extra cell wall enhancements, so to speak. Seeds are very tough. And so apoplast movement of the sucrose is happening. And again, if it's apoplast, I should, it's packed and sapped again, because you have to get back across the cell membrane at some point. And so primary and secondary active. So there's a bit of an energy commitment in doing any kind of apoplast sucrose. Here are these two, where I say sucrose, and sink, I'm just saying they can demonstrate that it's unchanged sucrose coming out of the sieve tube element, and it goes into the sink tissue as sucrose. Other sinks, it might get cleaved, where the sucrose comes out of the sieve tube element, gets cleaved into glucose and fructose, and then those get taken. So there's kinds of strategies. We don't have time to get into all the different strategies. So just be aware, it really, really depends on the variety of, or, you know, which sink are you talking about? And then it can be a variety of methods. So the next couple slides are just trying to demonstrate that. So here they're showing the companion cell sieve tube element is kind of a bluish green, and then the more olive green is looking at your parenchyma cells usually, okay? And so here they're showing apoplast because there's no plasmata right away. So here they're saying it could be apoplastic right as you come out of the sieve tube element, or maybe it's not apoplastic till a little further on in the parenchyma cells. So this is just, don't get lost in the details here. It's just to try to convince you that there's many strategies, many methods when you come to unloading, okay? All right, so you have all this sugar transport going on, and it can be, you know, different sieve tube element highway systems can have it moving in different directions. So you can have two sieve tube elements in the same, you know, vascular cluster, and one might be going down movement down to the roots. Another one might shift it more lateral, come off and do a more lateral or upward movement. So it just depends. From source to sink. You have to keep thinking source to sink. And it's not this unidirectional flow like what we see with water in the xylem. So once it gets to the sink, what are they doing with it? Well, again, depends on the sink, right? So they can use the photosynthate actively to stay alive. Like I need to have sugars so that I can make my ATP, so I can stay alive and functioning. So respiration is the shorthand for that. They might say, you know what, some darn bunny rabbit came along and chewed on me, and now I got a wound I gotta fix. And so they might need cell maintenance and growth. Maybe they're getting ready to start making a flower bud and grow and develop, and so they'll need that photosynthate. Or they could say, I have a pretty decent amount, I'm gonna stuff some away for storage and just save up for a rainy day in case some point in my life here I start getting low on sugar. I have some stored sugar to call upon. And so storage, and certainly with a potato tuber, that's really its main job. It wants to store up all the lovely carbohydrates, because if we don't eat that tuber, then, you know, in the spring, it'll sprout a new potato plant and use that tuber tissue as a source of energy for the developing plant. Of course, be aware, source is also doing it. The source tissue also has to stay alive. It also has to do cell maintenance and growth and storage. So the source is very important because it has to do all of that, plus the job of figuring out where do I send my extra sucrose. So it's constantly listening to other parts of its body saying, where should I send my extra sugar? And the sink is constantly saying, me, pick me. All right, and you have this kind of competition going on, and we'll talk a little bit about that next, actually. So what we have is that also, remember, it's not a set in stone thing. So you can have sink tissue morph into source tissue. And probably the best example of that is a baby leaf developing into a mature leaf. That would be an awesome example. And so it depends on its physiological state. Is it a net exporter or a net importer? And that will determine if it's source or sink tissue. And also, of course, like, you know, the developmental state. Is it a baby leaf or a mature leaf? That kind of idea. And so they've done some really nice, pretty straightforward experiments where they look at labeling the CO2 again with radioactive carbon, and then feed that into the, you know, around the plant and as they're doing photosynthesis. So what this is showing is that this is on the left is a baby leaf that is a net importer. And so at this time, when they took this picture during that part of its life, it was still a baby leaf going, I need all this sugar, give me sugar, give me sugar. It couldn't make enough of its own because it was still a baby. Didn't have all the photosynthetic machinery up and running. Now this is the same leaf over different time periods. As the leaf is growing up, so to speak, it starts to shift from being a net importer when it's all black. It's taking all that radio-labeled carbon from, you know, another mature leaf that did all this and sent it. Now this leaf is going, oh, you know, I'm doing better. I can start to shift now and become more of a source tissue because I'm growing up. I have all my photosynthetic machinery optimized. Everything's good to go. So this is showing you a visual of a leaf that initially was a sink. It was getting a lot of photosynthate from mature leaves probably. And then over time, it started to give back and give sugars off. And it was a net exporter to another baby leaf nearby, or maybe it was a baby leaf bud that was forming. This is the same type of experiment with, I believe this was a Arabidopsis, where instead of radio labeled, which isn't fun to work with, they used a fluorescent protein instead. And again, just trying to show how the source leaf, you know, took up the labeled carbon, and it's starting, you know, sending it off. And this leaf here is starting to transition into this one. So as the leaf grows up, it can shift from sink to source. Okay. So I want to introduce this term, photosynthate allocation. Please. So there's gonna be two terms. We'll start with this next time. Allocation and partitioning. They have very definite definitions in this class. Unfortunately, the way we use these words out in the real world differs, and that is confusing. Please memorize the definitions I provide and ignore what you've learned out in the real world, okay? All right, so allocation is looking at how the photosynthate is being used. So if you think about a source leaf and it's doing all that photosynthesis, the source leaf has to decide, what should I do with the sugar? Should I save some of it up? Should I use it for respiration? Should I use it for cell maintenance? Or should I transport it? So it's saying, how should I allocate all this lovely sugar I've made? How should I divvy it up? All right. So it's the metabolic fate of carbon. Allocation, it's really the metabolic fate of carbon. What are they going to do with that carbon? Respire? Build a new cell wall? Maybe they have a wound, store it up. Okay, so allocation means metabolic fate. All right, and then next time I'll contrast that to partitioning, and we'll describe a little bit about that.